All right, so now we're going to start talking about metabolism in cells specifically. We're going to start by talking about the beginning of something called cellular respiration. We're going to see in a second that cellular respiration has many parts. And so today we're just going to discuss the first part of that, which is something called glycolysis. Okay, brief overview of the whole process before we dig into it is that cellular respiration is the way that living cells get energy from organic molecules. So you eat a piece of bread, that bread has some starch in it, has some glucose in it. Our goal is to make energy that a cell can actually use out of that piece of bread. And the form of energy that cells can really use is ATP. So we're going to make ATP. We're also going to make our electron carrier NADH, which we talked about in our last video. So NADH is a reduced electron carrier. We're using it to basically shuttle electrons to our final step of cellular respiration. They're going to help us make some more ATP. Okay. When we're talking about cellular respiration, we're going to see that there are different types. Specifically, we'll see that there's aerobic cellular respiration and anaerobic cellular respiration. Aerobic cellular respiration, like when you're doing aerobics, right? Aerobic respiration uses oxygen. So if a cell is doing aerobic respiration, or if it's doing your normal type of cellular respiration, it's really our baseline. We're going to use oxygen and we're going to release CO2. So that's why humans breathe oxygen. You have to breathe in oxygen in order to help you break down all the food you ate last night. And then you breathe out CO2. That's a byproduct of this process that we're going to be talking about. Okay. Primarily, when we talk about cellular respiration, we're talking about the breakdown of glucose. Other organic molecules can be used. So you can break down fats, you can break down proteins, you can break down other things for energy. But those aren't your cell's preferred way to get energy, and not all cells can do that. So we're thinking glucose. So our overall chemical equation for glucose metabolism, which is going to be aerobic cellular respiration. So aerobic respiration, glucose metabolism, basically refer to the same thing. We're referring to the fact that we take a glucose molecule. So glucose is C6H12O6. We remember that carbohydrates always have that format, that ratio of carbon to hydrogen, oxygen. 1 carbon to 2 hydrogen to 1 oxygen. Okay, so that's glucose, C6H12O6. We add in oxygen. We actually add in 6 molecules. Okay, we're going to get out carbon dioxide and water. So after this whole process, all that you have to release out into the world is carbon dioxide and water, right? So you don't see any more carbohydrates, no new carbohydrates at the end here, just CO2 and water. Okay. So this is pretty neat. And we're going to think about where all the energy from breaking these glucose bonds go and how we get ATP out of this process, because we'll notice that it's not in that equation. Okay. As we go through glucose metabolism, the process is pretty complex, so we break it into parts. So we can think of it as 4 parts or 5 parts, because our 4th step kind of has 2 parts. Okay, so first, we're going to have glycolysis. So that's what we're going to talk about today. Glycolysis is going to end up with this molecule called pyruvate, which we're then going to break down. Our end product from pyruvate is going to get shuttled into something called the citric acid cycle, or the tricarboxylic acid cycle, or the Krebs cycle. Those are all the same thing. And then finally, we're going to send all our electron carriers to something we call oxidative phosphorylation, which has two parts. It has an electron transport chain, and it has ATP synthesis. So that's just the whole process that's going to happen. I've highlighted on here where ATP actually gets made, because if you take nothing else away from this discussion of metabolism, I want you to absolutely know where ATP gets made, what steps in this process produce ATP. So I'm front loading that right now. The places we're gonna make ATP is we're gonna make a little bit at the beginning in glycolysis, and we're gonna make a little bit more in the citric acid cycle. Almost all the ATP we make from aerobic cellular respiration is going to be at the very end of this process during ATP synthesis, during oxidative phosphorylation. So as we're working through all of this, our goal is to understand what's happening with our electrons. For each of these steps, what carbon-based molecule do we start with and end with? We want to think about any important additional pieces that we might have to add in. We'll specifically see that we need some coenzymes as we go into the Krebs cycle, TCA cycle, citric acid cycle. Most importantly, we want to focus on the amount of ATP that is produced by each of these stages. Here I've listed out the stages you should be thinking about: glycolysis, breakdown of pyruvate, otherwise known as pyruvate oxidation, the TCA cycle, which is the Krebs cycle, which is the citric acid cycle, electron transport, and ATP synthesis. I really love this diagram, and I'm going to tell you why. It's because what you're actually looking at here, that swirly bit, that pink bit in back that you kind of can't quite see, that's a mitochondria. So this picture is actually showing you where all of this is happening inside of a cell. Okay, so we see our steps here. Here they've broken them down into 4. So we see that cellular respiration starts out with a glucose molecule outside in the cytosol. Handy dandy label here, cytosol. Okay, so that's where we're starting. We'll see that what's happening here throughout the process of metabolism is that we're actually moving a bunch of our products into the mitochondria. We move it across 2 membranes, across the outer and the inner mitochondrial membrane. We move our products here into the center of the mitochondrion, which is what we call the mitochondrial matrix. Okay, so here in the mitochondrial matrix, we have a lot of the rest of our process going on. And we'll see also in a little bit that a very important part of the way we make ATP is that we build up a gradient in this space between the outer and inner mitochondrial membranes. So this is called the intermembrane space, INTERMEMBRANE. Okay, so the intermembrane space is going to be really important as we get to the end of this process. So as you're thinking about the steps of metabolism, I want you to be very careful about thinking where we are in the cell and where we are relative to the mitochondria. This is super important if we're thinking about cells generally, because we know some cells don't have mitochondria, right? Eukaryotes have mitochondria, but prokaryotes don't, right? So any part of metabolism that has to happen inside a mitochondria has to look different if we're thinking about a prokaryote. Okay, so glycolysis is happening out in the cytosol. It's not in the mitochondria yet, so it can happen in any type of cell under any condition. But these other steps are only going to happen in specific types of cells. So just a little additional note about how to read this diagram. So what it shows you is it shows you carbons. So these little black circles are carbons. So it shows you your initial carbon-containing molecule. So in this case, glucose. And then your product. So here we can see that in glycolysis, glucose is going to get broken down into smaller carbon-containing molecules, 2 pyruvate molecules, each of which will have 3 carbons. Okay, so that's one thing it's showing you, is the beginning carbon molecule and the end carbon molecule. We can see it does that for each step. Okay. It shows you how many NADH, so reduced electron carriers, are being produced. So we can see on this flow chart, we have our NADH coming here from glycolysis. We're making some. You can see that we're making some in these other steps as well, right? So in step 2 and step 3, we're making more. And it shows you that they're all going to this final step, oxidative phosphorylation. And this diagram also shows you where you make ATP. So it shows you a big bang at the bottom, big explosion when you make some ATP molecules. So it's really tracking all the things that we want to track. So if you're going to focus on something, you want a nice, concise picture, this is a good thing to focus on. We're going to focus on just the glycolysis part now. So we're going to go into this in a little more detail. I know in your chemistry classes, it's possible that you go over all the different enzymes and all the intermediate products. We don't need to do that here, but we do want to understand the basically fundamental way that this is happening. So I'm going to talk about it in a little more detail than you necessarily need to know. Okay. Important things about glycolysis. It starts cellular respiration. Even though we need oxygen for aerobic cellular respiration, we don't need it at the beginning. So glycolysis is not the part of this reaction where we use the oxygen. So glycolysis can actually happen without oxygen. So glycolysis is going to happen in a cell that's in an environment that has lots of oxygen, but even if your cell runs out of oxygen, you can still do glycolysis. So that's super important because sometimes the only place your cell can get energy is from the energy produced during glycolysis. Okay. So glycolysis happens whether or not there's oxygen. It's happening in the cytoplasm. The steps in glycolysis are pretty much identical in all living species because this doesn't require mitochondria. Okay. There are 10 total steps. We're going to think about them as 3 sort of conceptual phases: an energy investment phase, a cleavage phase, and an energy liberation phase. So here we see a picture of this reaction. So we're starting with our glucose molecule. So we see that in blue. And above it, we're tracking the carbons. So those black circles, again, are carbons. So we're starting with a 6-carbon molecule. So we want to think about the number of carbons. So we have a 6-carbon molecule, glucose. During the energy investment phase, which is steps 1 through 3, so here in the first little third of this process, what we do is we actually put in some energy. Remember that this process, this beginning of glycolysis, is not energetically favorable. We talked about that in our last video. So we have to put in some energy right at the beginning of glycolysis in order to make this product called fructose 1,6-biphosphate. It's steps 1 through 3, so there are some other intermediate products in the middle there. We're not concerned with them right now. Okay, so basically, the reason we're doing this in glycolysis is we're trying to make a molecule that's going to be easier to break apart. But in order to make this easier to break apart molecule, we actually have to put in some energy. So we put in 2 molecules of ATP. We use up 2 molecules of ATP in order to create this first intermediate, this fructose 1,6-biphosphate. So we've made no energy so far. We've actually lost energy at this point in glycolysis. But then we move into our second phase, cleavage. So we don't get new energy yet. What we do is that we break our 6-carbon molecule, in this case, fructose 1,6-biphosphate. Don't worry about that name, but do know that it's still a 6-carbon molecule. So we have our 6-carbon molecule. We started with glucose, which was a 6-carbon molecule. Now we're taking a 6-carbon molecule and we're chopping it in half, right? Like with a cleavage. Okay. So we're breaking it in half into 2 3-carbon molecules. Okay. So that's our cleavage phase. Steps 4 and 5, we break our 6-carbon molecule into 2 3-carbon molecules. Next, we have our energy liberation phases. So that's steps 6 through 10, aka the rest of them. And steps 6 through 10 are where we actually produce products that we're going to want for our cell. So during the energy liberation part of glycolysis, we break down our 3-carbon molecules into our final product of glycolysis, which is pyruvate. So these 2 final molecules are pyruvate. Those you do want to know the name of because they're going to come back in our next step in our pyruvate oxidation. Okay? So we've gone from glucose to pyruvate. So that's during energy liberation. It's called energy liberation because this is the part of glycolysis where we make the ATP. So as we're making this pyruvate, we release 2 molecules of ATP for each 3-carbon molecule. So that's a total of 4 molecules of ATP. So this can get a little tricky as we go through metabolism because we've split our 6-carbon molecule into 3-carbon molecules. You're going to have to pay careful attention as to whether we're focusing on just 1/2 of this pathway or whether we're focusing on both parts of this pathway. It's better to think about both together because when we sum up how many ATP are made, how many electron carriers are made, we really want to think about it from a perspective of for each glucose molecule, so for each molecule we started with. Okay, so in total, as we get to our products, we make 4 ATP molecules. And we also produce 2 molecules of NADH. So at the beginning of glycolysis, we actually had to put in some NAD. So we started with some NAD. Now we're going to add in some electrons. So we're going to tack on some electrons onto those NAD. They're now NADH. They're now reduced. These are electron carriers. We're going to take those electrons to the end of our process.